Assalamualaikum warahmatullahi wabarakatuh. I am Izan Afisa binti Muhammad Ruzaili and I will be presenting to you my final year project entitled Isolation and Identification of Lignocellularity lactic acid bacteria from rumen sample. Based on the background of this study, lignocellularity is extracellular protein that could be characterized into ligninolytic and hydrolytic enzymes. Since purified enzyme is costly, microbial enzyme used as alternative in industrial applications. Lignocellulose is a part of plant biomass composed of cellulose, hemicellulose, and lignin as shown in diagram 1. The problem statement behind this study is about the large amount of lignocellulose waste will be underutilized and resulted in landfill, which could lead to environmental pollution if they left undegraded. Besides, fossil fuels can also lead to global warming, hence alternative bioenergy resources are required. Next, with the benefits of lignocellulose degradation into simpler form of monosaccharides, alternative method to non-renewable resources could prevent environmental issues. We move to the objective of this study. First, to isolate lactic acid bacteria from rumen sample for identification of lignocellularity activity. And second, to identify the isolates using microbiological and molecular method, which is PCR. Regarding Chukwuma et al. 2020, bioconversion of lignocellulose biomass, it involves pre-treatment, enzymatic hydrolysis, fermentation, product separation and purification. Since bacterial strain grow faster than fungal strain, they have ability to produce lignocellularity enzymes. According to Saini et al. 2021, actinobacteria is one of the significants in lignin degradation. Calderon et al. 2021 stated that lignocellularity enzymes can be applied in bioethanol production, biopulping and biobleaching of wood pulp. The methods during the experiment are: first, rumen sample was isolated into a mice transfer medium, then tenfold sterile dilution followed by propate method on MRSA gas supplemented with bromostressor was done. After 2 days incubation, the bacterial colonies were further isolated onto CMC, which is carboxymethylcellulose agar, with 3 days incubations. Next, the isolated colonies were further isolated on MSM, which is minimal salt media, with lignin extract agar with 7 days incubations. The cellulose activity on CMC agar was screened with congorate, while the lignin activity on MSM lignin agar was screened with methylene blue. Biochemical tests such as gram staining, catalase, motility, sulfide and indole test, and also MRVP were done. Molecular methods such as DNA extraction, PCR and purification were performed to determine the species and genus of the organisms. And gel electrophoresis was run and visualized. The result obtained from the isolation of bacterial isolates on MRS agar, where the bacterial colonies morphology show small, mucoid and circular white colonies in figure 1. But the colony on MRS agar with bromostressor green showed greenish color at the center of the colonies, and the color of the agar was green. Next, congorate was used onto CMC agar to determine the cellulose activity by degradation zone. But the absence of degradation zone indicates lactic acid bacteria from rumen sample was not produced cellulose as shown in figure 2b. Lignin activity was screened with methylene blue, and the absence of degradation zone as in figure 3b showed that lactic acid bacteria from rumen sample was not a lignin degrader and have not been reported yet. In addition, the bacteria exhibit purple color due to the presence of thick peptidoglycan, indicate gram-positive bacteria with Coccobacillus shape. Catalase test showed positive result with bubbles formation. However, lactic acid bacteria do not require oxygen for growth and not produce catalase. This may be a false positive result due to human error or contamination. The bacteria show negative result in motility, sulfide and indole test, where no red color formation in indole test and no formation of black precipitate indicates no sulfide production. Methyl red negative bacteria will metabolize fermentation product by decarboxylation, and no acetone production by the bacteria resulted on yellow color of solution as in figure 7 and 8. The extracted DNA of bacterial isolates have size of 10,000 kb. The expected apicon size of bacterial isolates with universal primers 27 forward and 1492 reverse is 1400 bp. The obtained PCR bands size were 1200 bp, and the bands located at the top has larger fragments. The brighter bands were due to high concentration of DNA. Next, size of purified DNA obtained was 1500 bp, which was higher than PCR products. The size of PCR bands were smaller than purified PCR could be the large amount of DNA, which will lead to slower migration. The presence of smeared bands may be due to high DNA concentration or contaminated with protein. In conclusion, the objective of this experiment to isolate lactic acid bacteria that was expected to degrade cellulose and lignin were successfully screened. The bacterial isolates were not a cellulose and lignin degrader, hence no degradation zone occurs on the selective agar plate. The microbiological tests and PCR were done to identify the specific species of the lactic acid bacteria. However, the species was not determined yet due to the waiting of sequencing result at this moment. For recommendation, in future, research may be conducted by isolating the bacterial samples from cellulose or lignin sources. The cellulose and lignin activity should be measured quantitatively to evaluate the amount of cellulose and lignin present in the mixture. That will help to detect the degradation activity. That's all from me. Thank you.